extremely low given all that it does. And our challenge is the following, that is there any hope at all in our understanding how the brain works and functions and develops in a situation where this organism in many ways eludes understanding still in a predictive manner. This is a bacteria, E. coli, many of you are familiar with that, and it, like the brain, receives all sorts of inputs, senses sugars, carbon sources, temperature, pH, and so on and so forth, takes this information and responds to it by moving its flagella. And we've got an enormous understanding of how each of these signals are processed by this bacterium and how it responds, but we still don't have predictive capacity to understand how this works in quote-unquote real life while the bacteria is around. How does it respond or not, and what are the ways by which such multimodal inputs are integrated, analyzed, and result in response. So in a situation where we know rather little about one small cell and how and what it can do, is there any hope at all about being able to decipher how 86 billion cells work together? Now, the answer to that will come up in my talk, but basically, if you break up complexity into its units and ask how those units are made, there is some hope of understanding. There's another aspect of the brain, in addition to its complexity, in that it is something which we as humans, our brains, we as humans are concerned about, is about our well-being, and the brain is something which is affected, particularly as large populations in the world age, with disease in a way which is peculiar and distinct from diseases which affect other organs. It's not that they're intrinsically special diseases, but if they affect our capacity to think and understand and move, then they have particular consequences which other diseases may not have. Here is a brain which has been infected with a tapeworm, causes a disease called neurocystic fibrosis, and this is something whose diagnosis was made possible only by the invention of CAT scans and MRIs and so on. Until then, people who had this disease had epileptic seizures, and people didn't know why they had those seizures. Epilepsy can be caused by many parameters, and only post-mortem would reveal the actual causes. So this is an example of how technology could be used to diagnose and treat diseases at early stages which are otherwise not accessible. There are other diseases which probably are lifestyle diseases, diseases such as Parkinson's disease, Alzheimer's, dementias, which are affecting a larger and larger number of our populations all over the world, and their understanding is also as important as their treatment. So the brain is something which we would like to know about, how these 86 billion cells are put together, how they function, and how we age, and how the brain functions in health and illness. Now, in order to understand such complexity, we really ask three kinds of questions, and these are the questions which biologists ask no matter what topic they're addressing. And these three questions, which have been summarized very well by Sidney Brenner, are: where does it come from? And that's evolution. The second question is, how is it made? That's developmental biology. And the third question is, how does it work? That's physiology. And no matter whether you're working on a plant or a bacterium or an elephant, if you're a biologist, these are the three questions you keep asking again and again. If you are a medical doctor, you also ask, how do I fix it if it's broke? So that's more of an engineering approach to biology. So let's go through this. Where does it come from? So very quickly, the Earth was formed about 4.5 billion years ago, and let's say that's that wall over there, and if you say that it's each billion year is 10 steps, then it's about 40 steps away. Now, the universe was formed 13.5 billion years ago, so even earlier. About 1 billion years after Earth was formed, life came about on Earth. And so 3.5 billion years ago, life came about. We know very little about the mechanisms of how that happened. There's lots of speculation, lots of work going on, and lots of theories. But let's assume for this talk that we're talking about unicellular organisms. So the bulk of life at that time, all of life were unicellular organisms. And even now, the bulk of life on Earth is composed of unicellular organisms. Over time, hundreds of millions of years, billions of years, you had different forms of life coming up. And that was really elucidated and understood by the work of Charles Darwin and Alfred Russel Wallace, shown over here. And they showed that its evolution takes place by natural selection to give rise to this wondrous beauty of different life forms all over the earth. And that's something which all of you are familiar with. So let's quickly run through this timeline and how it happened. So about 3.5 billion years ago, they had simple cells. Going on down to the bottom of the slide, multicellular forms of life came about 1 billion years ago. Then further dramatic changes took place. About 600 million years ago, simple animals came about. And then about 550 million years ago, complex animals. And there was an explosion of complexity around that time. And we still don't know how these extraordinary forms of animals came about in such a burst. And there are again many theories about that. And there were land plants about 475 million years ago, and 400 million years ago, insects and seeds were detected or came about. Much later, from 360 million years of amphibians, then reptiles and mammals about 200 million years ago, we have about 130 million years ago flowering plants coming about. So you can see that from 13.5 billion years ago of the universe starting, as it were, 4.5 billion years of Earth, it's only in hundreds of millions of years that we're seeing these dramatic changes in life forms, even though life originated about 3.5 billion years ago. Then something dramatic happened about 65 billion years ago, and those kinds of events can happen now too. And this was an asteroid crashing on Earth in what's now Mexico, and that resulted in the elimination of large animals such as land dinosaurs. The cousins still live amongst us, birds, but the big dinosaurs were all gone. That resulted in the potential being opened up for expansion of the mammalian radiation, and mammals started on their journey of essentially ending up with humans ruling the earth, as it were. So about 2.5 million years ago, we had the appearance of Homo, and 200 years since the appearance of 200,000 years since the appearance of modern humans, and only 25,000 years ago, some of our close relatives, the Neanderthals, disappeared. So this is really a dramatic change which took place because of an asteroid. Now humans have done amazing things, and that's because evolutionarily what has happened is that our ability to oppose thumb and forefinger, our ability to speak and process language and communicate have allowed us to evolve culturally at great speeds and create instruments which can not only be used to understand nature, but can be used to change nature. And this has had a runaway effect which is distinct from all other animals. And the human brain, which is disproportionately large to our body size compared to other non-human primate brains, is something which has had a major role in this. So these 86 billion cells, neurons are there, which is much more than that of any other animal, and certainly much more than that for our body size as compared to other primates. And so that's a big debate about how that happened, how did evolution select for these kinds of changes, and so on and so forth. We still don't know, by the way, whether our brain, we have more neurons than the African elephant, but people suspect that experiment has not yet been done, but people suspect that African elephant which has a larger brain probably has a larger brain because of more glial cells and not because of more neuronal cells. So let's go on to answer our three questions. Our first question we've answered, which is about how did it evolve? And this evolution of this brain, by the way, is not only at the macro level I've talked about, but also of its complex. It's important to remember that even though this looks extraordinarily complex, one of the principal simplifying features is a shared chemistry right from unicellular organisms to now. And therefore, understanding the chemistry of the brain and understanding of the chemistry of any cell in our body or any other organism is feasible by studying any other cell in any other organism because of the shared chemistry. And the shared chemistry is linked by the thread of DNA, and therefore the genetic material becomes a very strong proof of evolution, but also a very strong instrument of our understanding of the mechanisms by which these changes come about. So let's go on to the next question about how it is made. And to answer that, there are really, again, conserved mechanisms which apply to all of animal development, which are simply stated and which are very complex in their actual application in each context. And that's something which everyone tries to understand, whether you're studying how limb development takes place, or heart development, or fin development in fish, or brain development. Basically, cells start off, for example, as a fertilized egg, divide to give rise to 2 cells, 4 cells, and as they do this, they become distinct from each other in terms of the potential they have to change, to differentiate into specific kinds of cell types, such as arms and legs and so on and so forth. This differentiation comes about by 3 ways. One, cells can divide to pass on to their daughter cells components from the mother differentially. So one daughter gets something and the other daughter gets something else, and therefore by a lineage-dependent mechanism, cells can become connected with each other. Cells can also be in contact with each other, and by contact-dependent communication, they can influence through signals how one cell becomes different from another. And cells can also have communication at short and long range. And so, for example, if you take a group of cells from one location and put it far away, if it were transforming its larger area by its location, putting it else will allow it to transform that area and have a long range. So, short range communication, long range communication, and lineage are 3 ways broadly by which cells become different from each other as they develop. There's a 4th way in which cells also use very much in animals in particular, and that is as they differentiate, they also migrate over long distances. And there are rules for, again, interaction of the mind. So as far as the brain goes, this again happens, and there's decades of work on this. This is a picture of a cell about to divide, in which on the green side, one daughter will get one set of products, and on the red side, it will divide, and the other daughter will get the other set of products and become different from each other. And the brain is made typically by such cells, which are stem cells, dividing to give rise to another stem cell. And instead of having 2 identical cells as daughters, it will give rise to one stem cell and one other cell, which will then divide to make nerve cells. So stem cell gives rise to a stem cell and nerve cells, and stem cell divides again to give rise to a stem cell and another nerve cell. So that's illustrated over here. You have 𝑁V, which is called a neuroblast, which will give rise to another neuroblast, another neuroblast, another neuroblast. Each neuroblast is different from each other, and each neuroblast also gives rise to what's called a ganglion mother cell, which gives rise to 2 neurons. So you're starting to manufacture nerve cells by this recurrent division of stem cells. Now you do this across the axis of the animal. You do it from head to tail, and you've defined head and thorax and abdomen and different tail segments. And you do it from top to bottom, and so you have different parts of the brain from the dorsal to the ventral, and you do this process again and again. And because each specific location has a label, each of these stem cells make nerve cells which also carry that label. And in a spatial manner, they make nerve cells appropriate to that location, brain or spinal cord and motor neuron and so on and so on. This is the broad principle. Vertebrate cells do it somewhat differently. This is in an insect cell, but essentially the principle is this, with vertebrate cells also migrating in large numbers and having different kinds of cells. So these kinds of changes in spatial division of cells is also seen at the molecular level, and at different times you will have different molecular signatures of this division taking place, so that we know how different neurons which are born at different stages in stem cell divisions are different from each other. So in a spatial manner, you can pack a particular segment, each segment across the axis of the animal, different from each other with different kinds of neurons, and layer on this, there's one more difference, which is you can have temporal changes, as you can see here, that the molecular markers change with time, and different kinds of molecular markers come about and shut down. So spatial changes, temporal changes, and axis location help define how cells are made. And this is something, by the way, which happens to non-neuronal cells also. This happens to non-neuronal cells also, and this is the fundamental principle by which diversity is generated during development. Now, this results in something which we are all aware of, and that is regional specialization, because the control along the top of the animal to the bottom in the brain here, shown over here, or left to right, or middle to outside, results in different parts of the brain being defined as different from the other. Optic lobe, the olfactory lobe, areas which are processing memory, motor systems, and so on and so forth. These also become different. At the core of this whole process, again, we must keep in mind are fundamentally the cells. And these cells are very, very important, because in the brain, the nerve cell, for example, is a cell which receives inputs from hundreds, thousands, millions of other cells, and then takes those inputs. These nerve cells take these inputs, transmit them along their axon, and send signals to their target cells, which again they connect to many, many cells, as I've told you about. So this fundamental unit of the nerve cell is something which is there in every part of our body, and these nerve cells fall into the following categories. They can excite the downstream target cell, they can inhibit the downstream target cell, or they can modulate the downstream target cell or groups of cells by making them more excitable or more inhibited. So just as there are fundamental rules of making nerve cells, there are also rules about how nerve cells end up functioning. So this is a picture of a stem cell, showing the big cell there and showing out all the other progeny which are coming. And the result of that is the regional specialization I told you. Stem cells are not only the ways by which nerve cells are generated, but groups of stem cells, a lineage defines a functional unit, and those neurons related by lineage also are related by the function they do. So this group of stem cells here, for example, will give rise to a set of cells in the olfactory region of the adult fruit fly, shown over here, and perform a certain kind of function. And there are many, many other lineages which will also altogether function as a unit. So this is a very interesting and important simplifying feature. You have taken an extraordinarily complex brain, which has, first of all, a simplifying feature of the nerve cell being common, and there are different kinds of nerve cells. Then groups of nerve cells are born together and they have similar kinds of function. So you actually have a functional simplification through lineage, and different lineages in a particular region function to, for example, in the antennal lobe, will function to do a particular kind of task, and that again allows another kind of simplification. So regional simplification, simplification because of lineage and because of cellular function allows us to look at these complex structures. Now, what I've told you in this case for the fruit fly is broadly true at the molecular and cellular level, and also in the level of circuits in many cases, about how circuits are put together, how there's cells involved in the circuits function and the molecules function across many animals. So by studying one animal again, you can understand the other animals also. And this is a picture made by my friend Benny Shilo illustrating this. And basically, that just as I told you, those mechanisms of making cells different from each other by lineage and by cell-cell communication, at the molecular level, there are only a few pathways, a shared toolkit which is used to make brains such as ours, as well as brains such as fruit flies or any other animal. And these are also used to make other tissues in us. And these toolkits really function through molecular pathways, and those conserved pathways are familiar to many of you. They are the notch signaling pathway, the hedgehog signaling pathway, the wind pathway, and so on. Just a handful of them. So these toolkits are used again and again in a context-dependent manner, spatially and temporally defined, to result in the diversity which we see. So we've learned about where the brain comes from through evolution, how it is made, and now let's go to how it works. Now, how it works was a very big challenge and continues to be a challenge in new ways. But those kinds of questions were addressed really elegantly some time ago by electrophysiologists. And one example is the work done by Hodgkin and Huxley. Hodgkin and Huxley took a nerve cell, a giant neuron from a squid, and squeezed out the insides of that nerve cell and put back in fluids of known composition and poked an electrode inside and outside, poked an electrode across and poked electrodes at different times by varying the composition of the extracellular and the intracellular ionic medium, and saw how electrical currents or action potentials, similar to what happens in real neurons, propagate from one end of the nerve to another. This is extraordinary because they could mimic how action potentials function. They did that first. And then by replacing the fluid with defined composition, they could decipher the way this happened. And that they wrote down a set of equations which showed how the action potential could propagate from one end of the axon to the other. Now, these equations are very interesting and important. They are not just a model, a mathematical equation which describes the action potential, but those parameters actually predict the existence of particular kinds of ion channels opening and closing to allow this to happen. So this is a beautiful example of a forward approach as opposed to a reverse approach. I'll come to that at time, of deciphering what the molecular properties should be, and then later on people went and discovered these channels. So this is absolutely exciting. Now, just as we can look at the physiology of one channel, there's been dramatic advances in our technology, mainly using optical tools of various kinds to look at physiology, as distinct from electrophysiology of this kind, which allows us to look at the activity of groups of neurons connected to each other. So we can examine the activity now of hundreds of neurons, in fact thousands of neurons, and this shows two neurons in the basolateral amygdala, work done by researchers at MIT, and they found that these neurons play a very critical role in separating positive and negative experiences. So this ability to move from recording single cells to that of being able to record hundreds and thousands of cells gives us an idea of what is happening in brains as they function, because you can do this in live brains, and you can do this in both freely moving animals in some cases or tethered animals in others, and see what happens. And we can do that in humans in much cruder ways, much cruder ways, by doing magnetic resonance imaging, for example. So these studies tell us the importance of regional specialization and how information comes into different parts of the brain is apparently integrated or looked at from different other kinds of inputs at higher centers, and these layering of different centers of the brain to process information and resulting in output is something which many people are studying intensely, and I'll come to that now. So we've covered how it is, where it comes from, evolution, how it is made, developmental biology, and how it functions, physiology. But really, what do we know about how the brain works? We know that you get a huge number of afferents coming in. Something seems to happen, this question mark is, and then you have an output. So what is happening inside? Of course, you see nerve cells firing. They talk to each other because they fire together, or stimulated by the other, or inhibited by the other. Can we ever hope to understand how all this means, what all this means, and rather than just a description? Well, there are many theories about how the brain works, and some talk about there being higher and higher centers which actually decipher, understand, processes information, and take decisions. And others talk about just as a whole lot of information being passed from one place to another, and a judgment call being made that, okay, in response to this kind of input, I will either run away or stay, and then next time onwards, you use feedback from what you've learned before, because your nerve cells are different from what you've learned, and you calibrate the next experience and make a next kind of guess. So humans and many other animals, all other animals, are really very good at making educated guesses about their environment. And people have speculated that this is very valuable, because if a deer were to say that, look, I see spots on a yellow background, black spots on a yellow background, let me sit down and analyze what it means. It will be eaten up by the leopard. It just has to assume that these black spots on a yellow background means a leopard and just run. Right? It could be wrong. It could be someone's bedsheet lying there, but that's something which you cannot wait and analyze. So making educated guesses from past experience or from very small numbers of experience is a trait which neural circuits have developed again and again. And that's illustrated in this. So you can't hear the audio.
Canaletto has this fantastic detail, and you can get right up, really just get right up and see all the details on the painting. And I started across the hall in North Carolina because I thought it was probably a Canaletto and would have all that detail. And I noticed that the bridge there's a lot of people just barely see them walking across the bridge. And I thought as I got closer, I would be able to see all the detail of those people, see their clothes and so forth. And as I got closer and closer, I actually screamed. I yelled out because when I got close, I saw the detail wasn't there at all. It's just real clear.
So this artist has basically not painted people on a bridge, but just put blobs of paint on something which looks like which on a bridge. And because you assume that people should be on the bridge, you have in your head deciphered specific kinds of people carrying things, walking, laborers, others, and so on and so forth. But it's just little blobs here and there. So this is how the brain seems to make guesses and work. And it takes all sorts of inputs and takes decisions in split seconds in a way which is pretty impressive. So how that happens in such a distributed structure, and how is it that it's correct so often, is an area of intense research. Now, how do you research that? And here is another movie. This is, if you can see that, a fruit fly, and it's about to land on these cylinders. And of these 4 cylinders, one of them has got banana paste on it, and the other doesn't. And this fly is smelling, it's looking at the air, the temperature, hovering around, and it will make a decision integrating all the inputs, including how hungry it is, what day of the week it is, and so on and so forth, and make a very precise landing on the wrong cylinder. So why did it do that? We have no idea. You know, I mean, there's got such a huge blob of banana in the other one. It's hungry. It's supposed to go and land on that and eat it, and it went over here. Now, to understand this, what scientists have done is they are now able to look at the behavior of individual animals at large scale, not just by one camera looking at one fly, but cameras looking at many animals running around in a social context doing things. You can also open up the brain of the fly, and very recently what people have done, and this has been done for the maggot of the fly, but it will soon be announced for the adult fly. The maggot has a few thousand nerve cells, and people have mapped by electron microscopy sections the alignment of all these 3,000 nerve cells with each other and how they connect to each other. So the connectome of the maggot is known, and the connectome of the adult fly, of which you're seeing one nerve cell, will be known soon. And so we will know, unlike just a regional specialization, a very high resolution regional specialization of the brain of the adult fly. Not only that, we can genetically activate and inactivate any neuron of your choice, and we can do that without opening up the animal, so we can see how it affects the behavior, and you can do this in many animals or one animal as you choose. Not only that, you can open up the animal and make RNA or look at other metabolites, at gene products from the animal, and have a profile on every nerve cell of what is the chemistry of that nerve cell. So you can know what genes are expressed, what proteins are expressed, what metabolites are expressed, what neurotransmitters are expressed, and how that neuron affects the behavior of the animal when it's activated or inactivated. This might allow us to get a better and better description of how circuits function in real life to elicit behavior. Now, this is very nice, but it also poses a aesthetic problem, particularly for people such as physicists. But is there a general theory on how the brain works? Now let's step back a little bit and ask about how genes are involved in the development of animals or in our function. Genes, as I said, encode components of our toolkit which are used to make us. But which toolkit is deployed where is a consequence of regulation of that toolkit. The regulatory part of the toolkit is not genetically encoded. The executive function comes as an interaction of that regulatory region of a gene with the cellular environment and the external environment and the world outside. So how that executive function is put in place to cause gene expression at a particular time and place needs to be understood in each and every cellular context, and therefore big data and big ways of looking at it allow us to understand. In a similar manner, neuronal cells are the fundamental unit of the brain. They form circuits. These circuits interact with other circuits in the brain in different kinds of layers, and how those layers are interacting with each other in an executive form to take decisions about the environment is something which also needs to be understood and described on a high scale, and that is again feasible. So it's about looking at algorithms and how they work, and not about a theory of the brain and how it is solved, and even higher function such as consciousness and so on can come. So if you want to get another metaphor for this, you can look at this termite hill on the left, and that termite hill can be seen as some kind of an inverted inside-out brain. You have termites building this complex structure. Each termite, as a very poor analogy, would be likened to a nerve cell. It receives inputs and it executes an output. It doesn't have an awareness that it's building a hill. It doesn't have an awareness that all of this is happening. Cells get inputs, analyze it, pass it on to other cells, but somehow there's this emergent property of an extraordinary hill. And we're getting a better and better understanding of how such emergent physical properties of swarming and development of this kind take place, and we will also have a better understanding of emergent properties of how different complex behaviors come about. On the right is a cathedral in Barcelona by an architect. That has a central command. Antoni Gaudí decides that he wants to direct the construction of this kind of a cathedral, right? And that's different. So human brains have the capacity to talk to the environment, to other human brains, to machines, and have the central command kind of structure. But we have evolved from a biological brain which has this distributed kind of emergent property. The result of that is what we have done is we have the ability not only to use our brains better through language and communication, we've created machines which can start to think in rather dramatic ways. I don't have time, I won't go into that, but in 1950 Alan Turing asked the question, can machines think? And Turing was famous for many areas of work, but the answer to that question seems to be yes. Soon there will be a seminar here, a few years from now, in which there will be machines present here listening to the talk, and we won't be able to distinguish which of the sleeping people are machines and which of them are computers. So there are going to be dramatic changes which will happen in artificial intelligence, and artificial general intelligence, which remains to be a challenge, will start becoming something potentially very real and something which we should be concerned about. So machines, according to Stuart Russell and Peter Norvig, will have to be called thinking machines if they can think humanly, think rationally, act humanly, and act rationally. And those kinds of machines are something which it doesn't seem so unreal now as it did a few years ago. So this kind of an artificial intelligence where we can design machines to make these structures are going to be very rapidly feasible. These are descriptive structures which we can make machines do. But independently thinking machines which can use this information, process it in a manner similar or distinct from the way our brains do it, and respond to that are also becoming real. The energy use of such machines is not 20 watts, it's hundreds of thousands of kilowatts. But that's something which is a different aspect question which needs to be addressed. But machines certainly will become more and more like us in very interesting, complex, and difficult ways. So thank you very much.
If you have any questions, there would be volunteers who will pass on the mic and then we can ask the question.
Yeah, there's a hand raised at the back.
Hello, sir. I'm a third year student from the physics department. So you spoke about brain cells interacting with each other and with other levels of the brain. What I understand is brain is basically made up of nerve cells which conduct electricity. So what do you mean when you say they interact with each other? Is it just passing on electrical impulses or is it something else?
So typically, nerve cells, neurons, receive an input which are chemical signals through what are called neurotransmitters. Then that signal is processed inside the nerve cell and results in a change in the membrane potential locally, which results in the creation of an electrical signal which is transmitted from one end of the nerve cell to another. At the other end, again, this arriving electrical signal is used to create a chemical signal to send a signal across to a connecting another nerve cell. So it's chemical signal, electrical signal, chemical signal. And this happens in trillions of connections. This is a typical way of stating it. There are lots of exceptions and a huge number of different cells.
And this seems very doable artificially. I mean, receiving some chemical signal, changing the potential, and then sending an adequate response to that potential.
It's a very crude and slow process, and electrical circuits can do it much, much better.
Yes.
Right? They do it at much higher energy. So equivalent electrical circuits which mimic nerve cells and mimic memory were some things which were started decades ago, and they've gotten better and better. But to actually mimic in the laboratory an actual creation of a nerve cell signaling has proven impossible for any kind of cell, for a variety of complicated reasons which deal with these kinds of phenomena coming from hundreds of millions of years of evolution, and they're not easy to recreate in a stable manner in the laboratory.
The follow-up question would have been, if it is doable in a laboratory, then the human brain is capable of having new ideas or understanding new concepts. So can we expect AI or other artificial made equipments to understand new concepts rather than depending on a database to?
Yeah, short answer is yes. I mean, even though machines would be constructed in very different ways, they have some broad shared principles about different layers and recurrent feedback mechanisms and so on and so forth. But yes, broadly, yeah, it should not be unexpected to think about it.
Sir, you mentioned about the efficiency of the brain being around 20 watt, and similarly if we with current machines that we have being in hundreds of kilowatts, like what do you think, even if at all we know, is the reason for the such orders of magnitude difference in the efficiency of the machine that is today?
Well, the whole way brains and all our tissues work is through analog mechanisms and through squishy, dirty chemistry. How that results in something so efficient it still remains elusive from the point of view of energy. So a mechanism by which inefficient use of energy available in a distributed manner, but on such a large scale, making trillions of connections, can extract information which is robust and stable is fascinating. So we don't know that. The converse is true of the way the machines we build. We build them in a digital way, largely, in ways which are extremely efficient, but energy very, very expensive. And therefore that requires scale, memory, computing, and energy at a level which human brains and many other animal brains don't need.
So even if at all we try to understand how it works in the human brain case, as you mentioned, we could take a layer by layer scan in case of as we did for a fly. Similarly, when it is alive, is it possible for us to get any idea in that region by doing such operations?
We can do an analysis of how animal brains work in live animals at a very fine level of granularity. We can't do that in human brains for obvious reasons, but because of both disease, which I didn't have a chance to talk about, and damage, there are many human conditions, both at the molecular level, at the cellular level, at the level of circuits, which tell us a lot about how human brains work.
Good evening, sir. Like you talked about the termite's nest and the castle made by a human engineer or an architect. So regarding that, the termite's nest can be thought of as emergent, like you said. Like a single termite cannot perceive it. So would you regard anything displayed by human society as emergent, like which a single human cannot perceive, but human society builds? Is there such an example with human society?
Yes, yes. There's really nice books about how scale and emergent properties of scale come about. I think the name of the author is 𝒉𝒆. That book's name at least is Scale. But there, this question is addressed about how human populations, cities, for example, how they come about, how their organization is in many ways actually similar to the internal organization of a termite nest. How is that a shared emergent property? How do groups of people leave rooms and come about? So there are, of course, proximal interactions and signals of various kinds. But collectives have certain kinds of underlying physics which many people have studied very well.
Hello. Good evening, sir. Yeah. Great talk, sir. So it's about memory. My question is about memory. So, like 2-3 years old kids don't really retain memories from their childhood as adults, right? So, what is the reason behind that? I mean, as adults, we tend to remember most of our life, but when we are 2-3 years old, we do not remember anything from that part of our life. So, what is the reason behind that?
So you said adults remember?
I mean, right now I could remember if I'm like what happened 10 years ago, right? But I cannot remember the things that I did when I was 2 or 3 years old.
Do you remember what you did day before yesterday?
I mean,
No, I mean, this is a serious question. So I think this is a very interesting point. So what seems to happen is there are two components. One, the fundamental of sensing and memory, and we know quite a bit about it, not enough, of course. But there are phenomena which are inhibitory to the consolidation of memory or of their recall, which sets in. Now, these inhibitory phenomena are very, very important. The reason I asked you about whether you remember something you did day before yesterday is if you recall the time when you're growing up as a kid, if you walk down a street, you would remember the name of every shop, where it is, the next shop, what song was playing, and everything. Now you don't even notice that walking by. The reason is that the ability to forget, to inhibit, and not to be able to recall is something which happens as the brain matures. Early on, that ability to inhibit is not there. So the inhibitory signals are put in place a little later, and they get strengthened by the way we use and what we do in most people. Some people still have amazing recall, but they are very, very dominant because it appears that if you don't have those inhibitory signals and inhibitory neurons acting, those memories will constantly be coming back and your day-to-day function will be completely gridlocked. So inhibition is not about not having the memory, but it's usually about not recalling it because of active inhibition.
Good evening, sir. What is the from endocrine gland and brain? What is the relationship? Like, is there any pineal gland or anything like that?
So, I mean, this question keeps coming up, particularly in relationship to higher function and such as consciousness and so on. That there used to be this view that the pineal gland was something which was a seat of something or the other. Increasingly, there is no, or rather, there is data to suggest that there isn't any particular seat of consciousness, as it were. So, understanding consciousness requires the testing of many different kinds of theories, but the pineal gland as a seat of, or endocrine glands as a seat of that kind of higher learning doesn't seem to be the case. If you want to understand consciousness at a level of some rigor, I would suggest you to look at a Nature genetics paper by Anil Seth and a colleague that looks at the various theories of consciousness and how they can be tested and which are more likely to be testable and which are untestable. And he's also written a book.
Sir. Okay, sure. Yeah. Thank you so much, sir, for your talk. It was really illuminating. So my question is a bit oblique. Do you think, based on the talk that you've given us, do you think that we're looking at bacterial infections and antibiotic resistance in a wrong way? Do you think we should revise our the way we look at it and look at it from a point of view of microbial communities rather than just one particular organism? What is your opinion on it?
This is like asking a chemistry question in an astronomy exam. I'm in deep trouble. Um, yeah, no, I think bacterial resistance is very, very important. And of course, with respect to the brain, there are infections which are very important. But that's a long topic which we can discuss separately.
So you had mentioned about certain behavior.
Where are you? Oh, sorry.
So you mentioned about how certain behaviors develop. So, for example, we have certain behaviors such as dietary switching in animals. Those are things that are genetically passed on. They don't have to learn it from their parents. But then there are certain behaviors that,
So certain behaviors like what? What did you mention?
for example, dietary switching behavior.
Right.
Yeah. So those that is genetically passed on. Like even if they don't have a parent, they know, okay, during some particular time period they can choose between two kinds of nutrition. But then there are certain behaviors that have to be learned from their parents. So is it that during evolution, after a while, something can become like, for example, some behavior that had to be taught by a parent can become genetic afterwards, or are these always two independent entities?
No, I don't think you can generalize, but even if you look at a very important point you said about dietary switching, and many insects do that. So, for example, the female mosquito will feed on sap, but before egg laying will feed on blood because you need a high protein diet for the development of the eggs, and that's the selective pathway which is chosen, and the parasite has jumped onto that. But when push comes to shove, this is not a rigorous program. Alternatives will suffice. If there's no blood at all, or there's no sap at all, other things will do. Male mosquitoes don't do this, but male mosquitoes can be forced to try and feed on blood, even though they can't feed on human blood because of the way their proboscis is engineered. But they will try to do that. So there are, behavioral switches which are substantially have a large genetic component, but multimodal inputs, like you saw with the fly landing on the wrong place, play an important role. So typically they will go to an attractive smell, but for other reasons they can move around. That's something which is very exciting, which is happening now. We're trying to understand.
I must thank you for your nice lecture, but I admit I was sometimes yawning at that time. Anyway, my question is that you mentioned about the stem cell. What do you think about the future of the stem cell in the treatment of neurological disorders, particularly damaged liver or from injuries? If you can.
Now, this is an important point. Um, there was a phase when one could take stem cells from a person, reprogram them, and the idea was that these reprogrammed cells which were taken, made into stem cells and reprogrammed, could in multiple ways be used for regeneration for tissues including such as the spinal cord and the brain. Now, our understanding of spinal cord regeneration, our understanding of nerve regeneration and damaged sets of neuron regeneration has improved dramatically in mouse, in zebrafish, and to some extent in fruit flies. But our ability to engineer this to any level in human has not worked out well at all, including by using stem cells. Stem cells have worked really extremely well in certain human diseases such as blood diseases, where stem cell therapies have proven to be very, very effective. I won't go into that right now. But they have not yet been effective in these kinds of situations. This area of intense research, both in terms of treating dementias, but also in treating, for example, spinal cord injuries.
Good evening, sir. My question is, we see that twins have very different personalities. Sometimes, even if we are very closely related to our parents, we have a very distinct personality. So, how do you think that is encoded?
when we see we genetically share a lot, environmentally also we share a lot, but what comes down to us having very different personality as such.
This goes to the core of the title of the topic of today's lecture about nature and nurture. And while I talk to you about how the connectome of different organisms is being deciphered, to assume that having genetic identity between different flies, for example, or a very similar connectome means that you will have stereotyped behavior no matter what is not evidenced at all. There's a huge variability. So there is a variability in the connectome, and even developmentally for genetically identical animals, there is enormous variability in the nature of development. At the risk of taking a little more time, so here are genetically identical animals, right? And look at the enormous range of differences in size. I mean, they're completely identical. These are ants. And these are ants which have got different roles in the nest, and they're specialized to be different depending on the role. And so therefore, interactions with the environment during larval development, either by food or by other kinds of cues, have resulted in these ranges coming. This is the queen in that nest, in this army ant nest. Sitting on top of the queen's head is a genetically identical, essentially, worker ant. So what you eat in humans, what you're taught, what books you read, where you live, where you are brought up, all of that have a phenomenal impact on what happens. And so it's not genetics alone that's important, that puts certain boundary conditions. But you must realize that culturally, humans are very different from ants in that we have developed prosthetics of various kinds over centuries which allow us to function no matter what our genetic strengths and limitations are. So if I'm hard of hearing, I can get a hearing aid. 100 years ago, if I were hard of hearing beyond a certain level, I would be consigned to be considered stupid because I cannot take part in conversations, and I descend into being further and further away from those conversations. Similarly, if I can't see well, I can wear spectacles. If I can't understand something, I can Google it. And tomorrow, if I can't understand something, if I have a problem writing an essay, I can go to ChatGPT and get that done. Right? So this has constantly resulted in us being able to transcend the limitations of our genes and genetics. Right? Flies can fly, but humans cannot. But we can make an aeroplane. So yeah, there's a huge impact of the environment, not only in this way, about what we eat and so on, but culturally that impact is disproportionate. So opportunities are what make us different from each other or similar to each other.
Good evening, sir. So it was a nice talk. So my question lies in the cathedral, Barcelona cathedral, and that termite hill situation. So, like emergent traits, they are shared across some organisms, and there is some chemistry behind like why a certain trait arises in a variety of organisms. So, has there been any study regarding why a certain kind of structure is being preferred across some organisms as an emergent trait?
Yeah, so that people have started looking at it in many different contexts. It's not that every single group of termite makes that kind of a hill, right? There's a lot of termites which make mounds which fail and flop and so on and so forth. There's spacing between each other and what happens, and sometimes something clicks and this goes off on various kinds of self-organizing development of this hill, which has a certain limitation about its size. It doesn't grow infinitely. So we know the proximal chemical interactions in many cases, but we don't know the emergent properties which feed back into the level of density of the population, the environment, the heating, the cooling, what decides when to stop.
Sir, great talk. So I just wanted to ask. So let's say hypothetically we know the human brain's connectome, right? And so what kind of information can be obtained from that, and what kind of information can't be? So
Yeah. So the maggot of the fruit fly, that connectome, took from start over a decade of work by several laboratories. People are now trying to look at the connectome of a small cube in the mouse brain, which is 1/500th the size of a mouse brain. That will take decades too. A mouse brain will take centuries, and a human brain might take several centuries at the current pace. But there's dramatic change in the technology development for getting EM slices and aligning, and in terms of segmentation aligning all the data which is taking place. So in a manner analogous to how DNA sequencing has progressed to unimaginable speeds unimaginable 30, 40 years ago, you're going to see dramatic change. So in theory, it is possible to get connectomes. So the connectomes will be of two kinds. This connectome of the maggot was a one maggot. It would be great to have many maggots, many mice, many humans, and then you would see how that relates to their lifestyle, their behavior, and what they live, right? And see whether there are any dramatic differences. We'll also try and understand what are the ways by which different neurotransmitters act in different regions, where different connectivities act to give rise to different behaviors, both in pathology and in diseases and in normal contexts. Um, It is, I would say, if there's an analogy, I would put that with astronomy, where understanding basic theory and principles in astronomy one does to a huge extent, but better and better telescopes in space and on Earth looking out have given greater and greater detail and better description and understanding. It's not merely description, but it tells us how things came to be. That's what you will see with human.
Good evening, sir. I had a question. So I am not a biology guy, but I was curious to know that we know that what distinguishes the AI from the humans is the ability to think. We have the ability to think. We don't need a predefined set of framework to choose from. So neurobiologically, if you can make it clear that what is something that that triggers the process of perception building, the process of thinking in our brains, the perception building particularly.
Yeah, so I mean, this was a view of some years ago that humans can think and have perceptions and ideas and so on, where machines cannot. That has changed dramatically, both because of our understanding of how the brain works and how machines work. It's quite possible that human brains are actually not thinking, as in, there is no center for perception saying, okay, now I have perceived this, right? It's just that you make a guess, you get multiple kinds of inputs, make a guess, land at the right place, land at the wrong place, use that information the next time. So it's a mush inside, and it's all of this is our first is an approximation which we create, and machines on the other side, they actually are getting more and more information, and based on that, they are taking decisions which again we don't understand how these AI machines work. The pace of change of AI is so rapid that the amount of effort which goes into understanding how the machines work has not been sufficient, because the machines change so rapidly, and the input, output, and the weights. So the way that works is that they give weights to outputs and boundary conditions, and the machine does some work and gives you predictions. Right? People haven't investigated enough what happens inside, except we know that there are certain kinds of layers, and those layers have recurrent loops and feed forward and feedback and so on and so forth. But how machines do to get this idea is something which will be understood soon. But about how humans take information and understand is something which may not be that different. So there may not be a perception center. Thank you.
Sir, yeah. Sir, you mentioned about the Parkinson's disease in your slides. Sir, like what I know is Parkinson's patients, they're under constant medication. So, like, is there any permanent cure for the Parkinson's disease?
So the challenge with treating any disease anywhere in the body is this problem of this evolutionarily shared chemistry I told you about. If you have a problem in one part of a body which is due to excess or lower production of a chemical, the activation or inactivation of a chemical, you can design a drug which inhibits that or activates that, let's say. But the problem is that you now administer that drug not to the specific region or to the specific cells which are affected. You administer it to the entire organism. And therefore, all the other cells which use this pathway will also be affected. So, unless the disease is so severe that you don't mind the side effects, the side effects of this approach are going to be substantial. And in many cases of severe Parkinson, the disease is severe enough that you don't mind the side effects. There's another component which happens is cells don't like to be told externally what to do by simple switches, right? Because they are a complex interaction of multiple pathways, multiple chemicals, and tweaking one can, for some extent, change the status of a cell from one level to another, but cellular homeostasis will pull it back to that state for some variety of reasons. And that means there will be bypass pathways which make it resistant effectively either by direct mechanisms or by indirect ways to the drug. So that poses another thing. These are not problems which are going to go away, and therefore our treatment needs to take these into account as well as other things.
So far, there is no cure.
Yeah, but there are better and better kinds of drugs coming in and treatments and modulating that, which can address these kinds of issues in Parkinson's and other diseases somewhat, and significantly better than it could be some time back.
Sir, and one more thing, like in old age patient, old age person, so Parkinson is common. But sometimes, like young age, like mid 30, 35 or 40, they too are affected by Parkinson's. So, what is the reason behind it? Is it genetical or something?
Well, that's interesting. There are two kinds of broad reasons. One is that there could be familial diseases which manifest early on, right? And the other is that the disease could manifest early on for other kinds of environment reasons. So people are also looking for dementias of various kinds at behavioral correlates much earlier than the strong manifestations, whether it's schizophrenia or Parkinson's or other kinds of neurological illnesses or dementias. Are there early examples of behavior which indicate a potential danger? So with Parkinson's, for example, it would be if much before you show the symptoms of Parkinson, are people tripping and falling even though they're physically able? Are there motor defects of some kind? That might give an indication on what to do. Now, there are potentially interventions at early stages which can be more effective, but I'm not familiar with the area. I don't know how effective.
Yeah, so let us thank Professor Vijay Raghavan for giving us such a wonderful talk. Now I request Professor Sudhakar Panda, Director NICER, Shri Ashok Parija, grandson of P.K. Parija, Professor Usha Vijay Raghavan, and Professor 𝒉𝒂𝒊𝒏 to come upon the stage. I request Professor Sudhakar Panda to felicitate Professor K. Vijay Raghavan and Professor Usha Vijay Raghavan with a bouquet of flowers. Now I request Professor Sudhakar Panda and Shri 𝒉𝒂𝒊𝒏 to hand over the memento to Professor K. Vijay Raghavan. So I request all the members of the board very much to be kind of. So the first P.K. Parija lecture series, this talk has come to the end. It is our pleasure to thank Professor K. Vijay Raghavan for delivering this first P.K. Parija lecture in life sciences. We also like to thank Pran Krishna Parija Charitable Trust for this endowment, and we also like to thank 𝒍𝒍𝒏𝒆𝒔𝒔 Berhampur for hosting their part of the talk. I'd like to thank Director 𝒉𝒆𝒂𝒍𝒐𝒑𝒎𝒆, Dean, faculties, administration, finance and accounts section, transport section, library and auditorium section for arranging this particular event. And finally, I'd like to thank all the members of other institutions and 𝒉𝒆𝒄 for joining this event. So special thank to Professor Vedant Mohanty. And the tea is actually arranged outside. Please, everyone can come and join. And if you have further questions, you can then interact with Professor K. Vijay Raghavan. Thank you.